Chapter 5 in the Campbell Biology in Focus book covers membrane transport and cell signaling. So we hopefully understand now that our cells are enclosed or covered in a plasma membrane that separates that cell from its surroundings. So acids, bases, toxins, and other things are protected from what's inside of the cell by that plasma membrane. The plasma membrane is something called selectively permeable. This means that it is selective about what it allows to cross through, and some things are allowed to cross in easier than others. So think about like a guard gate at a neighborhood. A guard is going to be kind of the selectively permeable membrane there, allowing certain cars to enter who have the logo for the neighborhood, whereas it's going to stop other things, other vehicles, and make it harder for them to cross over. So selectively permeable means that some things can come over easily and others cannot. Um, so phospholipids are the most abundant lipid in most membranes. So most of the membranes are made up of phospholipids, and they're amphipathic, and that means that they have two parts to them. They have that hydrophobic tail and then that hydrophilic head. So they like water in some parts and dislike water in other parts. And remember, that's how it makes the two tails want to pop in and touch one another or get close to one another. And so we get this phospholipid bilayer. And that's one of the reasons, because there are parts of it that are hydrophobic, that make that cellular the phospholipid bilayer makes it selectively permeable. Sorry. So here's an example of our extracellular or our cell membrane here. So on the outside where it's kind of blue, that's the extracellular matrix. It's kind of just like the junk that's floating around our cell but isn't enclosed in our cell. And then the cytoplasmic side of the membrane is the side that contains all of the organelles that you learned about that cells have. And that cell membrane has a lot of stuff going on. It has lots of proteins incorporated with it. It's got that phospholipid bilayer. It has some proteins that are just on the outside. It has some proteins that go through the whole cell membrane. It has, and those are called integral because they're integrated. Peripheral are those on the outside. So there's just a lot that happens in that phospholipid bilayer. Our cell membrane is made up of a pretty complex thing. The fluid mosaic model is a model that we use that tells us that the membrane is a mosaic of protein molecules. They're bobbing in this fluid, and they're not stuck here. They're moving side to side. It's not a solid thing. We consider it a fluid mosaic model because it's constantly moving. And so you can kind of see this video kind of shows that fluid mosaic molecule. It's kind of bobbing and floating, and those little red things, those little proteins are moving in and out. So if you look closer at the bilayer, you have the hydrophilic heads of the phospholipids, those phosphate groups up top that really like to be around water, and then the tails, the hydrophobic tails, those carbon tails that really do not like to be touching water. And so they naturally go tail to tail, and head to head is out, and those are, that makes it a very tight, like chain link fence, because those tails don't want to touch water at all. They have to be forced to do it. Most of the lipids and some of the proteins in the membrane shift about laterally. That's why we call it the fluid mosaic model. And so the lateral movement, so side to side movement, is pretty rapid. Of the phospholipids, they're flipping places really quick, like dancers. But the proteins are bigger and bulkier, and so they're going to move a little bit slower. Some proteins move in a directed manner, like they have a direction that they want to go, and then others seem to be basically anchored in place. And we know that you get these proteins that like to move around because when a mouse cell and a human cell combines over time, it will all the proteins will be mixed together after an hour. So that supports the statement that these phospholipids are constantly kind of laterally moving. As the temperature cools, membranes switch from a fluid state to a solid state. So the colder things are, the more solid they are, just like with anything. Temperature at which the membrane solidifies depends on the type of lipids that are in it. And like for instance, if you have a lot of unsaturated fats in those phospholipid tails, right, then you're not going to solidify very much. Membrane remains fluid to a lower temperature if it's rich in phospholipids with unsaturated hydrocarbon tails, but they don't have kinks. Membranes must be fluid to work properly. They're usually about as fluid as salad oil, which is pretty amazing that they are so protective of the organelles inside of the cell, yet they're actually really flimsy. The steroid cholesterol has different effects on membrane fluidity at different temperatures. At warm temperatures, cholesterol restrains the movement of phospholipids. At cool temperatures, it maintains the fluidity by preventing things from tight packing. So, you can see in these, in the top two pictures, unsaturated versus saturated hydrocarbon tails. The more unsaturated tails you have, they can't hold as close together. There's too many kinks in the armor, too many kinks in the chain, and they can't be right hand to hand, shoulder to shoulder, hydrophilic head to hydrophilic head. Whereas when they're saturated tails, they pack as tightly as they can. When you add cholesterol to a membrane, you reduce the membrane fluidity, but low temperatures hinder solidification. So even when your temperatures are low, those cholesterol keep it from being packed too tight. One reason why cholesterol is not great in your diet, but it's an important part of your diet because it's really important in our cell membranes. So evolution of differences in membrane lipid composition, variations in lipid composition of cell membranes of many species appear to be adaptations to specific environmental conditions. Ability to change the lipid compositions in response to temperature changes has evolved in organisms that live where temperatures vary. So if you're going to live in a really, really cold place, you probably need to have a lot of cholesterol within your membrane, or you need to probably have a lot of unsaturated fatty acid tails, right? Because if you're always, always cold, then you're not going to be able to get that movement of proteins and nutrients and elements across that membrane border that's necessary. A membrane is a collage of different proteins embedded in the fluid matrix of the lipid bilayer. We saw a picture of that just earlier. Proteins determine most of the membrane's specific functions. I showed you that there were these things called integral proteins that are found that go through the inside of the lipid bilayer. Sometimes they span it completely, where they pop out on either side, and sometimes they're just on one end or the other. So integral proteins that span the entire membrane are called transmembrane proteins. They're integrated into the cell membrane, and they're transmembrane because they're on the outside and the inside. The hydrophobic regions of an integral protein consist of one or more stretches of nonpolar amino acids, often coiled into these little alpha helixes. Peripheral proteins are just kind of stuck, glued to the surface of the membrane, but fairly loosely. So this is an integral protein, and you can see the alpha helix, right? And it's also found, so it's also a transmembrane protein found on the extracellular side as well as the cytoplasmic side, so the inside and the outside of the cell. There are 6 major functions of these membrane proteins, and they're very, very important in just everyday life of a cell. So either they will transport things across the membrane. They're also important in enzymatic activity. They will send signals across the membrane, and that will be important in like nerve cells, sending the message that your hand touched something hot all the way up to your brain to tell your brain that you need to put your hand away. Cell-to-cell recognition, so recognizing things that are like one another. Intercellular joining, and then attachment of the cytoskeleton and the extracellular matrix. Extracellular matrix is just the stuff outside of the cell, and the cytoskeleton, right, is kind of attached to that cell membrane. And so so in order that things aren't just bouncing around everywhere, they have to kind of be like, imagine Jello and sticking an olive or a cherry inside the Jello. It's going to float. It can move around if you get it really hot, right? But for the most part, it's just sitting still in there. It has to be kind of cushioned. So that's what the cytoskeleton will do with the extracellular matrix. So here we have some pictures of different things like transport, and we'll talk about specific types of transport a little bit later in this chapter, and that would just be movement of things across the membrane from the outside in or the inside out, and either it happens on its own or you have to add energy in the form of ATP. Then there's we learned about enzymes in chapter 2, maybe 4, I don't remember. And so with enzymatic activity, right, these proteins are the enzymes that allow chemical reactions to happen faster. Signal transduction, so things will attach, passes a signal through, and then the inside of the cell or outside of the cell gets that message. Cell-to-cell recognition, so cells, like cells, will kind of combine and hold hands with other like cells, so you end up with a tissue. Then you have intercellular joining, so when you have multiple cells, right, all packed together and joined together, it produces a tissue as well. And then I explained this a little just a couple seconds ago, attachment of the cytoskeleton to the extracellular matrix so that our cells aren't just bouncing all over the place and getting bruised and beat up. Cells recognize each other by binding to surface molecules, often containing carbohydrates, on the extracellular surface of the plasma membrane. Membrane carbohydrates may be covalently bonded to lipids, and when they are bonded to the lipids, it's called a glycolipid, because glycogen is a type of carbohydrate, right? So glycolipids, it's a carb fat, or more commonly, the membrane proteins are bonded to lipids, and they're called glycoproteins. I'm sorry, the membrane carbohydrates are bound to the proteins forming a glycoprotein. So carbs on the outside, external side of the plasma membrane are going to be different among different species and individuals, and even just the cell type of an individual. Membranes have a distinct inside and outside face. So there's outside the fence and there's inside the fence. There's inside the cell, outside the cell. The asymmetric arrangement of proteins, lipids, and carbs in the plasma membrane is determined as the membrane is built by the ER and the Golgi apparatus. So this is just a picture of, if you remember from chapter 4, I believe it was, when we learned about the jobs of organelles, what things look like in the plasma membrane is determined by the ER and the Golgi. Okay, so if we move on to concept 5.2, membrane structure results in selective permeability, you guys are probably going to do some experiments with this in lab, where you're going to do some diffusion and osmosis projects. So basically, a cell's got to move stuff across cellular boundaries. There's no reason why things should not be moved back and forth. Trash has to be taken out, good stuff has to be brought in. You couldn't survive closed up in your house without doing anything, right? You would have to have a way, you have to have a door, a way to let things in and for you to get things out. Plasma membranes are selectively permeable, as we talked about, and they regulate that traffic of things across. It's like your doorbell and the eye hole, the peephole, regulate things, allow you to regulate things. The membrane itself regulates things. So the hydrophobic molecules, the things that are afraid of water, they're nonpolar, can dissolve in the lipid actually things that are hydrophobic, afraid of water, they're nonpolar, so they don't have a strong charge to them, positive or negative. They can dissolve in that lipid bilayer and just crush through it easily. Really easy for them just to come in and out. But polar molecules that are not hydrophobic, that are hydrophilic, and that have positive or negative charges that are fairly strong, they're not going to crush through pretty easily, because that hydrophobic tail of the lipid bilayer is going to stop it and tell it, I don't want you to touch me because I'm afraid of the things that you like. So we end up with these things called transport proteins, and they allow hydrophilic, loving water substances to cross the membrane. So some transport proteins called channel proteins have a hydrophilic channel so that certain molecules or ions can use the tunnel. And then channel proteins called aquaporins facilitate the passage of water. So we have transport proteins, which are probably going to be integral proteins, and transmembrane proteins, and they're allowing things to pass through, especially hydrophilic things to pass through. And if they're allowing water to come in, it's called an aquaporin. Other transport proteins called carrier proteins will bind to that molecule, change shape, and then allow it to cross the membrane. So it kind of tricks the lipid bilayer to letting it in. And transport proteins are specifics to the substance that moves it. So you will have lots and lots and lots of transport proteins along that lipid bilayer, along the cellular membrane, because there's so many things that have to come in and they're specific to it. Passive transport is diffusion of a substance across the membrane where no energy is invested, no energy is added to it. And so this is when it would be things that just come across on their own, or you have one of those transmembrane proteins that facilitate that movement, although energy is not added. Diffusion is the tendency for molecules to spread out evenly into the available space. Think about when you initially light a candle or put on perfume. Where you're standing or where that candle is the strongest in the whole entire room. If you were to spray your perfume in your bathroom, it's going to be very, very concentrated in one area. But over time, those perfume molecules are going to spread out throughout the bathroom and spread the smell out so that it's as strong in the corner, eventually in the corner of your bathroom, as it was in the spot where you sprayed it. Same thing with that candle. Initially, the smell from that candle is just right there by the couch where you have the candle lit. But over time, that smell is going to diffuse and it's going to be as strong everywhere, or should be, because diffusion is the tendency of molecules to even themselves out on their own. Although each molecule moves randomly, diffusion of a population of molecules may be directional. Think about your perfume that you sprayed. Your AC is going on. And it's going to move the smell with the wind. At dynamic equilibrium, as many molecules cross the membrane in one direction. At dynamic equilibrium, as many molecules cross the membrane in one direction as the other. So when we're in equilibrium, it doesn't mean that things have stopped. Equal is equilibrium. Things have not stopped. It just means as many are crossing over one side as are crossing over the other. So one membrane will lose two, but two will come in. It's equal direction.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
Okay, so if we use this example where it's molecules of dye, you put droplets of dye into water. Initially, it's very concentrated, right? And even if you don't stir it, that water will turn the color of whatever that dye is. So it's diffusing across the membrane. You have maybe some semi-permeable membrane here. So it's crossing over without being stopped. Eventually, it gets to equilibrium. It's moving from an area of high concentration to lower concentration until where you're at the point where the concentration is relatively equal, and that's equilibrium. Things are still moving across, but it's at equilibrium. And then here we have diffusion of two solutes. So the golden or yellow is in an area of high concentration, and it wants to move over to where purple is, to an area of lower concentration. It really doesn't even care how many purple molecules are over here. So you can see purple's in its high concentration on the right, wants to move over to the left, even though it's crowded with the gold molecules. Purple does not care that there are more gold molecules over there. It wants its own concentration to be spread out. And so you have equal movement of gold and purple moving across until the concentration is relatively equal. And now we're at equilibrium. However, we're still seeing some molecules crossing over. If we didn't have continual movement at equilibrium, then things would be dead. So substances diffuse down their concentration gradient. They move from an area from high concentration to lower concentration. No work must be done to move those substances. No energy has to be involved. So diffusion of a substance across a biological membrane is passive transport because no energy is expended. Passive transport, no energy. Osmosis is the diffusion of water across a selectively permeable membrane. Diffusion is just the movement of molecules, but osmosis is specifically for water. Back in the day, there used to be, I remember there was always an image of Garfield laying down with books on his head, and it said learning through osmosis. But in reality, that was incorrect because only water is moving in osmosis. It should have been learning through diffusion. Water diffuses across the membrane from the region of lower solute concentration to the region of higher solute concentration until the solute concentration is equal on both sides. So this is water moving only.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided. The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis.
Okay, so in this image, what we're seeing is, and this is like what you see all the time when you talk about osmosis and diffusion. So you'll probably see images like this on an exam. But this in the middle, this little dashed line, is a semi-permeable membrane. And so we have sugar molecules that cannot cross the membrane. Instead, water crosses the membrane in place of that sugar molecules. That membrane is too tiny to allow the sugar molecules to cross over. So in this case, this is an area of higher solute concentration, the solute being the sugar. This is an area of lower concentration on the left. So the water is going to want to cross from the left to the right to get to a point where we have equal, similar concentrations of the solute. This just explains that again. Now, when we look at water concentration and osmosis within our cells, we kind of are talking about tonicity. Tonicity is the ability of the surrounding solution to cause a cell to gain or to lose water. In an isotonic solution, the solute concentration is the same inside the cell as outside, right? So there's no net water movement across the plasma membrane. It's equal. It's equal water movement. That would be what's happening in this scenario on the right. Equal water movement. There's not a higher solute concentration. It's the same. A hypertonic solution is where the solute concentration is greater outside than inside the cell. And so the cell loses water. Hyper kids move around and leave all the time. In a hypertonic solution, whatever that cell is sitting in, there's a greater concentration outside of it than inside the cell. So the water wants to leave the cell to help the outside concentration get lowered. In a hippo, they don't move very much. They're really big. A hypotonic solution is where the solute concentration is less than inside the cell. So there's more inside than outside. Hippos are big. More water is going to enter the cell and make that cell get very, very large. So when we're in a hypotonic solution, for animal cells, it's not really a good thing. A hypotonic solution would mean that more water enters your cell and the red blood cells are going to pop. An isotonic solution is equal. A hypertonic solution is when the water leaves the cells and our cells are going to shrivel up. They're going to get smaller. So you know that when you get an IV, it's not pure water, right? It's a saline solution, a low solution of saline concentration. And that's because in our outside of our red blood cells in the matrix, it's a little bit salty. And so if you gave yourselves an IV of pure water, a lysing is what would happen. By allowing a little bit of saline concentration, we're equal with what's outside of our cells. If you had way too much salt in that saline drip that you got as an IV, you would end up with this hypertonic solution. So hypertonic and hypotonic environments create osmotic problems for organisms. So osmoregulation is the control of solute concentrations and water balance, and is necessary for us to live in different environments. I'm not even going to talk about him. And so this paramecium, I guess, has an adaptation to overcome that, where they have this little vacuole that will suck up water and hold it for when it needs it, and spit water out when it's got too much water, too little water leaving its cells. So cell walls help maintain water balance. Plant cells are different than animal cells. Animal cells are happiest in an isotonic solution, but a plant cell actually is happiest in a hypotonic solution. It wants to be a little bit swollen so that the cell wall is turgid, very, very firm. If your plant is wilted, it needs more water because it's in a lower solute concentration, and a higher solute concentration outside, so the cell's losing water. It likes to be in a hypotonic solution, so that keeps it rigid and solid, and so that's why even though they're flimsy little plants, that stem can be upright and the leaves can be nice and taut. If a plant cell and its surroundings are isotonic, there is no net movement of water into the cell, so the cell becomes flaccid and limp, and it may wilt. We got to water it again. If it's in a hypertonic environment, the cells lose water eventually, and the membrane pulls away from the wall, and you get usually a lethal effect called plasmolysis. So here you can see turgid is a happy way for plant cells to be, because that membrane, it's so full of water, that membrane pushes up against the cell wall. Remember that animal cells don't have a cell wall, but plant cells have a wall and a membrane. Well, if it's equal water coming in and coming out, that cell membrane isn't nice and tight. It's not pushing against the cell wall. And so it becomes flaccid. A plasmolyzed or plasmolysis is where the actual membrane pulls away from the cell wall, and this is really, really bad for that plant. Facilitated diffusion, passive transport aided by proteins. In facilitated diffusion, transport proteins speed the passive movement of molecules across a plasma membrane. So channel proteins provide corridors. They allow specific molecule or ion to cross the membrane. And channel proteins could be those aquaporins for help with water. It could be ion channels that open or close in response to some stimulus. So they're gated basically, and some stimulus lets it open or makes it closed, very similar to the stimulus from your gate opener, touching that, or the signal going to the box that tells it to open or close. So here's those aquaporins, image of the aquaporins. It's got an actual channel here that's facilitating the movement of these water molecules. Membrane and aquaporin. So carrier proteins undergo these little tiny changes in shape that translocates the solute binding site across the membrane. The shape may be triggered by binding and release of transported molecules. So that molecule might touch that carrier protein and make it move. And in this case, you don't have to require, there's no energy that's required. So this is all still passive transport. So here's a protein channel. It's just got a channel that's allowing things to just flow through, right? A carrier protein would be a molecule comes in, lock and key mechanism, and it pops it out on the other side. The only stimulus is this molecule entering this site and allowing it to be spit out on the other side, but we have not added any energy. When you add energy to things, that's called active transport. Active transport uses energy to move solutes across their gradients. Facilitated diffusion speeds transport of a solute by providing efficient passage through the membrane but does not alter the direction of the transport. Some transport proteins, however, can move solutes against their concentration gradient, so make things happen the way they absolutely don't want to do it. And when that happens, you have to add some energy. Active transport moves substances against their concentration gradient, so they go against the laws of diffusion. Active transport requires energy, and usually this is in the form of ATP, as all things in our cells and in our body need ATP to do stuff. That's our energy. Active transport allows cells to maintain concentration gradients that differ from their surroundings. And one very, very common type of active transport system is this thing called a sodium-potassium pump. If you get into biology or you take, biochemistry, or you take other higher biological classes, you'll learn all about the sodium-potassium pump.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is cotransport. Here, both sodium ions and glucose move into the cell through a cotransporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
This is just an image diagram of that sodium-potassium pump. So sodium binds to the sodium-potassium pump, and the affinity for sodium is high when the protein has this shape. When it binds, it stimulates this thing called phosphorylation. So ATP losing a phosphorus group kicks out energy. And then that leads to a change in the protein shape. So it spits out that sodium, kicks it out, and now has a different shape so that potassium can come in. And that makes that phosphorus kick off and get back onto making ATP again. And so it just kind of keeps going and going and going. So let's look at it again. We have 3 types of passive transport. We have 2 types of passive transport. We have diffusion, where things just move across the membrane on their own. We have facilitated diffusion, which utilizes transmembrane proteins. And we have 2 different types of facilitated diffusion, where there's just a channel protein, or a carrier protein. And then we have active transport, where you have to add energy, like in the case of the sodium-potassium pump, in order to move things across the concentration gradient that they don't want to. So they go against their concentration gradient. You always have to add water. In passive transport, you're moving things with their concentration gradient instead of against it. You're moving things that will naturally want to go that way. In diffusion, it happens on its own. In facilitated diffusion, it needs a little extra help because maybe these things don't want to touch those hydrophilic tails. So membrane potential is the voltage across the membrane, and voltage is created by differences in positive and negative ions across the membrane. So those ion pumps help maintain membrane potential. 2 combined forces collectively, called the electrochemical gradient, drive the diffusion of ions across a membrane. A chemical force is the ions concentration gradient, and there's an electrical force, which is the effect of the membrane potential on the ions movement. Basically, there's there's the electrochemical gradient that drives diffusion of ions across the membrane. An electrogenic pump is a transport protein that generates a voltage across a membrane. And the sodium-potassium pump is the major electrogenic pump of animal cells. The main electrogenic pump of plants, fungi, and bacteria is called a proton pump. I would just know what we use in animal and plant cells and how they differ, and that they are all called electrogenic pumps. And so these pumps help store energy that can be used for cellular work later on. So here's a proton pump. Protons are usually hydrogen atoms, pumping them across so that you end up with this positive and negative difference across the membrane. Co-transport occurs when active transport of the solute indirectly drives transport of other solutes. So plant cells use the gradient of hydrogen ions generated by that proton pump to drive active transport nutrients into the cell. So it's a co-transport. It's when active transport indirectly drives the transport of other things. So in this case, you have you have hydrogen pumping over. So you have the proton pump indirectly making that sucrose hydrogen co-transporter work. I don't care that you know what a sucrose hydrogen co-transporter is. I just care that you know what a co-transport is. All right, concept 5.5, bulk transport across the plasma membrane occurs by exocytosis and endocytosis. So when you have to get a whole lot of stuff to move across, we usually use these 2 things called exocytosis and endocytosis. Water and small solutes enter and leave the cell through the bilayer by means of transport proteins, but larger molecules, bigger things, have to be moved across in vessels, basically. And bulk transport also does require energy. Exocytosis is where the transport vesicle migrates to the membrane, fuses to it, and releases its contents. This is going to make a lot more sense once we watch some videos about it. So exocytosis migrates to the membrane, fuses with it, and then just spits its contents out. Endocytosis is when the cell takes in molecules by forming new little vesicles. So it's the reverse of exocytosis, basically. And there's 3 types of endocytosis, taking it in. There's phagocytosis or phagocytosis, which is cellular eating. Pinocytosis, pinching, cellular drinking. And receptor-mediated endocytosis. So let's watch some videos on these.
Endocytosis is the movement of materials into a cell via membranous vesicles. Exocytosis is the movement of materials out of a cell via membranous vesicles. These processes allow patches of membrane to flow from compartment to compartment, reminding us that a cell is a dynamic structure. Exocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract. In endocytosis, membranes invaginate, or pinch in, to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded.
What you should have noticed with the phagocytosis is that it reaches out, grabs it, and then closes off, which is going to be different from the pinocytosis.
In pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells and capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood. Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein-cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
All right, concept 5.6, the plasma membrane plays a key role in most cell signaling. In multicellular organisms, cell-to-cell communication allows the cells of the body to coordinate their activities. Communication between cells is also essential for many unicellular organisms. Eukaryotic cells may communicate by direct contact. Animal and plant cells have junctions that directly connect the cytoplasm of adjacent cells. And so these are called gap junctions in animal cells and the plasmodesmata in plant cells. So I'd know the difference, what each one is called, and understand that that's how they directly connect the cytoplasm of adjacent cells. Free passage of substances in the cytosol from one cell to the other is a type of local signaling, because sometimes certain cells are going to produce something that needs to be given to another cell. In many other cases of local signaling, messenger molecules are secreted by a signaling cell. These are called local regulators, and they don't travel very long distances. So one class of these growth factors stimulates nearby cells to grow and divide. And this type of local signaling in animal cells is also called paracrine signaling. So here you've got local signaling, paracrine signaling, where it's only going to signal something to very close by cells. And so you have this exocytosis, right? secretory vessels. They're secreting things that are going to hit the target cell and tell that target cell to do something. Another more specialized type of local signaling occurs in the animal nervous system, and this is synaptic signaling, consisting of electrical signals moving along a nerve cell that triggers secretion of neurotransmitter molecules. These diffuse across the space between the nerve cell and its target, triggering a response in the target cell. So this is, when we talk more about nerves, we're going to understand this a lot more, but this is synaptic signaling, where you have a specific target cell, and then you have electric signals that send out the message. In long distance signaling, plants and animals use chemicals called hormones. So like we have different glands that produce hormones, but it affects everything in our body. And so they have to travel long distances. It's not nearby cells. In hormonal signaling in animals, which is endocrine signaling, specialized cells release hormone molecules that travel via the circulatory system. So long distance signaling is endocrine hormonal signaling, and it travels via the blood system. I would know the 3 stages of cell signaling. So those 3 stages were discovered basically by this guy, Earl W. Sutherland, and this is on long distance signaling. And basically what he learned is that you receive reception of the stimulus, transduction of the stimulus, understanding it, and then responding to that stimulus.
Cells communicate with one another by means of chemical signals. For the receiving cell, there are 3 stages in the signaling process: reception, transduction, and cell response. The cell targeted by a particular signal has a receptor molecule complementary to the signal molecule or ligand. The ligand fits like a key in a lock and triggers a change in the receptor molecule. Signal transduction converts the change in the receptor to a form that can bring about a cellular response. This might involve a series of steps, a signal transduction pathway that alters and amplifies the change. In the 3rd stage of cell signaling, the transduction process brings about a cellular response. This can be any of many different cellular activities, such as activation of a certain enzyme, rearrangement of the cytoskeleton, or activation of specific genes.
Okay, so here are the 3 steps to cellular response. There's a reception, where you have a signal molecule and a receptor. It accepts it, and so it's receiving the signal. There's transduction. It transduces it. It understands it. It's like you taking in information, and then your brain kind of mulling it over, and then so it has these relay molecules that are sending this message along, and then there's an actual activation where something actually happens. Reception, the binding of the signal molecule to the receptor protein, is pretty highly specific because it's only going to accept what fits into it. So the signal molecule is the ligand, and the ligand binding generally causes a shape change in the receptor. Many receptors are directly activated by this shape change. Most signal receptors are plasma membrane proteins. Most water-soluble signal molecules bind to specific sites on receptor proteins that span that plasma membrane. There's 2 main types of membrane receptors. There's G protein coupled receptors and ligand-gated ion channels. G protein coupled receptors are plasma membrane receptors that work with the help of a G protein. And G proteins bind to the energy-rich molecule GTP. So like how we're talking about ATP before, this is GTP. Many G proteins are similar in structure, and the pathways are very diverse, though, in function. So you have your G protein, right? A signal molecule reaches it. It activates, the GTP activates the G protein and then activates this enzyme. So you get a cellular response. The ligand-gated ion channel receptor acts as a gate for ions when the receptor changes shape. When a signal molecule binds as a ligand to the receptor, the gate allows specific ions such as sodium and calcium through the channel in the receptor. Ligand-gated ion channels are very important in the nervous system, and diffusion of the ions through these open channels may trigger some sort of electrical response. So here you've got the ligand, the gates are closed, the signaling molecule hits this, the gates open, and then all of the ions can shuttle through. It spits out that signal molecule or the ligand, and it closes up that gate, changes its shape. Intracellular receptor proteins are found in the cytosol or nucleus of target cells. Small or hydrophobic chemical messengers can readily cross the membrane and activate receptors. So examples of hydrophobic messengers are steroid or thyroid hormones of animals and nitric oxide in both plants and animals. So if you're looking at hormones here, this aldosterone is a type of hormone that's secreted by cells in our adrenal glands, and it enters cells all over the body, but only the kidney cells have the receptor cells for it. So the hormone binds to the receptor protein on the kidney cells only and activates it, even though it's touching all the other cells and organs in our body. The active form of the receptor enters the nucleus, acts as a transcription factor, and activates the genes that control water and sodium flow. There's the image of that, and you can see that in your book also.